anatomy and function of different features of yeast cells. The purpose of this particular slide is to show you some details about the cell wall of yeast and some of the components that it contains, which are listed here and include chitin, for example. And so when I'm working with these guys in the lab and I want to crack them open to get a hold of their DNA so I can do some molecular work on them, I'll use an enzyme called zymolase, which specifically breaks down this component here, the beta-1,3-glucans, and rips apart the cell wall so that the relatively weaker plasma membrane can easily be disrupted by detergents, and then I can isolate the DNA from the nucleus. The yeast cell envelope, like I said, consists of a cell wall, which consists of a pretty significant amount of the dry mass of the cell. The plasma membrane is a lipid bilayer that's essential for selective permeability. And again, the purpose of selective permeability is to, as the name suggests, let certain things in and exclude certain things from coming in. The periplasm is where a lot of enzymatic activity exists. And if you look at the cell, here's the periplasm here between the cell wall and the plasma membrane. So this band here. And it's just like the periplasm of bacteria. And so again, as I said, there's enzymatic activity, and I gave you some examples here, like the enzyme invertase, which converts sucrose to glucose and fructose, and acid phosphatase that catalyzes the liberation of free phosphates from organic compounds so that the microbes can use the phosphates for growth. This slide gives you another representation of the anatomy, and it goes along with this table. So you can cross-reference things you see here with this particular table just to get a few words or a one-sentence explanation for what things do when you're studying for the exam. As far as bacterial growth goes, and even yeast growth, the typical stages of microbial growth in general follow these patterns. The first is a lag phase in which the microbes, when they're first pitched into a new environment, will gear up for growth in that environment by activating gene expression for the utilization of certain nutrients that are present or to deal with the environmental conditions. And once they've figured all that stuff out, gene expression turns on effectively and they start to grow exponentially. And eventually they'll exhaust their nutrients and waste products will be formed and the microbes will kind of nudge each other and antagonize each other, and they'll hit a stationary phase where they appear to not be growing, but they're certainly not dying. They're still metabolizing as best as they can, but in intense competition. And then eventually there's a decline or a death phase. And so again, what you're looking at is growth as a function of time. And here's just a micrograph representation of some bacteria starting off as a microcolony and then exponentially growing over a short period of time. So what's pictured here is asexual growth by yeasts. And so again, you can see if you look at the electron micrograph here, there's some bud scars in some of these yeasts where a baby bud had budded off and then popped out into the world and essentially did this process here. So here's our parent yeast, and here's the genome of the organism, and a bud begins to form from one of these areas where you see a bud scar, and then a new yeast cell is formed. And so you have a daughter cell here, and the daughter basically gets a full complement of the DNA from the mother. And then they're asymmetrical at first, but then the separated yeast cell will grow. Sometimes to approximately the size of the mother, and sometimes the yeast will remain connected to one another and form chains. And so here's another representation of that. Here's a budding event, this right here, and then you can see bud scars from when this mother has given birth in the past. And of course, this process that you see here continues as long as nutrients are present, like sugar or nitrogen sources or phosphorus, and as long as toxins aren't inhibitory. This slide represents sexual reproduction in yeasts, and you see the mating types, alpha and A type, that are haploid. So here's our alpha type cell and then our A type cell, and these are haploid yeast cells that have half the copy of the genetic information of a diploid. And so in this particular case, you see a sexual union here, or conjugation as it's called, in which the individual haploids change shape a little bit and then eventually fuse into in a process called schmooing, which is kind of dumb. I happen to have a picture of that. They call it because of this thing. I guess it's from an old cartoon or something like that. Anyway, um, so the schmooing event that you see here not only results in cell fusion, but it also results in fusion of nuclei. So they go from haploid to diploid, and that's this yeast right here that now has a diploid nucleus. And this guy can either continue budding in this process of that you see here, forming daughter cells that bud off of the cell, or if the nutrient condition is not favorable, they can undergo meiosis essentially and produce four haploid spores that you see here. So here's some alphas and then the A spores, and these guys can then go back and repeat this cycle and then eventually form another sexual union. Bacteria are just as sophisticated, but there's no sexual stage. So when I say just as sophisticated, I mean their asexual phase is just as sophisticated. So here you see a bacterium preparing to divide asexually by binary fission. And so the chromosome must replicate, and as the cell is elongating, so there's a lot of sophisticated stuff here, and I'm not going to get into the details, but somehow you need physical chromosomal separation, and then you need septum formation, and then full cytokinesis or cell division. And then you get two identical daughters that carry a full complement of the chromosome. And sometimes, again, the bacteria will stay stuck together and form chains. And so that's pictured here. You can see an electron micrograph of some bacteria dividing by binary fission. So in lag phase, as I said, the cells are synthesizing new components, and they have to replenish spent materials. They have to adapt to their new medium and temperature and pressure and salinity. And obviously the length of lag phase varies depending on the circumstances. And you can visualize that in a wine setting, where if you take yeast straight out of the freezer, for example, and pitch them right into a warm batch of a warm fermentation, a relatively warmer fermentation, the yeast will get shocked. In some cases, won't resuscitate at all. But typically, you have to slowly resuscitate the yeasts and then pitch them in, so that you don't encourage a long lag phase. The exponential phase is also called the log phase, and obviously the rate of growth is constant, and as long as the conditions are constant. If you modify the temperature right in the middle of exponential phase, you can create a second lag, for example, that would slow the exponential rise of the microbes. So again, the population is most uniform in exponential phase as long as you don't modify anything. And then stationary phase, the name reflects the fact that the total number of viable cells remains roughly constant. And again, it may occur because metabolically active cells stop reproducing. It may also occur because the reproductive rate is balanced by death rate. So there's deaths and some growth. The possible reasons for entry, some of the stuff I've already mentioned, nutrient limitation or changes in oxygen availability, certainly toxic waste accumulation. That's what we see in fermentation. Saccharomyces is a master of producing a lot of toxic waste in the form of ethanol. And so in some cases, it's just the critical population density is reached, and then the cells kind of communicate with each other that we need to stop. Various starvation responses are also interesting to study. They include morphological changes that microbes might make. So endospore formation in bacteria, a lot of soil bacteria produce these really tough, super resistant forms of life called endospores. And these things can obviously cause all kinds of sanitary problems in a winery, or they can get into a wine and potentially germinate under the right conditions and cause spoilage. But they're very difficult to eradicate, obviously, because they're extremely resistant to disinfectants and a lot of the methods that we use to try to get rid of them. Sometimes microbes can just decrease in size, and that's a function of protoplasm shrinkage, and they can also condense their nuclei. And then they produce some starvation proteins that help toughen the cell wall or protect their DNA under these harsh conditions. The goal is long-term survival. Sometimes microbes will even get nastier when they starve and and that's what we see sometimes in a public health setting where microbes that are starved increase virulence because some of the toxins that they produce are designed to steal nutrients from the host cell that they're infecting. So as far as morphological changes and decrease in size, this is just a little mathematical model, so you can see why being a big cell, your surface area to volume ratio in this particular example is 1.5, and if you just shrink your radius in half by a half, you can double your surface area to volume ratio. So this little cell in a nutrient poor environment would have an easier time, if my cursor is nutrients, getting that nutrient into the center of the cell as opposed to this guy where it would have a lot more effort to make sure that nutrients are freely diffused throughout the cytoplasm. Again, if you're looking at the formulas here, surface area is 4 pi r squared, so that's the numerator here, and the volume is 4/3 pi r cubed, and so this is a cube root function. So it shouldn't surprise you how how these variables are related. So this is just showing you survival strategies in microbes. So here's our pool of microorganisms under some physicochemical stress. And so an example might be sulfiting and how it affects Brettanomyces physiology. And so microbes can sporulate, for example. So bacteria can produce spores if there's some kind of stress. And I'm only going to emphasize the ones that are important in wine micro. And I already showed you how Saccharomyces and other yeasts can also produce spores. Just as I showed you in that sexual reproduction slide. But some microbes undergo a VBNC state, which is a really interesting thing that I'll come back to in a minute. So this slide just represents just under the microscope, just so you can see a chlamydospore. I didn't highlight this because Candida species don't tend to be particularly important in wineries, but this is a really interesting form of spore that some yeasts make called a chlamydospore, but that gives these guys, like Candida albicans tends to be a pathogen and can cause yeast infections in your mouth and urogenital tract. But they produce these spores when conditions aren't great or when they're exposed to antifungal drugs. So death phase has two alternative hypotheses. One of them is programmed cell death, where microbes run out of nutrients and then a fraction of the population is genetically programmed to die or commit suicide. And hopefully that makes sense why some of the population would, because those dead cells would pop open and leak their nutrients out, and they basically serve as food for their siblings, which is kind of weird. But so again, it's sort of an altruistic response. Whereas some cells undergo a viable but non-culturable transition, where the cells are alive, but they're dormant. And this is an interesting thing. We see this in a lot of different microbes, and it presents itself in a lot of different ways. So viable but non-culturable can be a good thing or a bad thing, but often in the food and wine industry, it's a bad thing because the microbes in this particular condition are difficult to detect. And so if you have VBNC microbes that are in your wine, for example, and you send your samples out for testing, and they don't grow on some of the tests that are used to detect them, you might get a report back that says, oh, your wine is fine. And sure enough, a couple weeks later, you get, some weird spoilage compounds, and the microbes were missed because they were VBNC, and the methods that the testing lab used were inappropriate. And I'll teach you some of those ways around the VBNC concept. You can also imagine this in the food industry, where individuals, if you have you're producing some food product or something like that, and you've got a bunch of VBNC salmonella in there, and you use the traditional techniques to try to detect salmonella in your product, and you don't find them, and then you ship your product out into the world, and then the microbes, after they're eaten, are still metabolically active and still can injure or kill people. So it is a neat concept, and it's one that's drawn a lot of attention for research. what microbes do in their VVNC state. And so this is just showing you a graph and illustrating the what's happening in like prolonged stationary and death phase. And so again, lag phase, nothing much is happening. There's an exponential growth, then a stationary phase, and then the microbes start to die. But this doesn't proceed to zero, right? And that's what this graph illustrates. Again, you're looking at cell viability as a function of time. And so you end up in this prolonged starvation state. And if you hone in on this and actually take samples from a flask, for example, of these, this could be a fermentation, for example, and where if you take samples of juice from that fermentation and test it for different kinds of microbes, you might actually see, instead of a straight line like this, you may actually see, if you use the right tools, changing populations. So GASP number 1 is a group of organisms that are undergoing a process called growth, advantage, and stationary phase. And so this is a GASP group 1, where these guys have figured out that there's some nutrient in here that they're really good at using, and they'll quickly bloom and use it up and then drop off. Whereas GASP number 2, this group then can maybe eat some kind of a waste product that these guys put into the medium, and so on. So it's a lot more dynamic. When you look at it at this level, it just looks like kind of a slow, steady state of, Saccharomyces or whatever. When in fact, if you look at it at a closer level, using molecular tools and things like that, you may find that it's just overlapping populations of different microbes rising and falling in the extended fermentation